Chapter 9, Cellular Respiration and Fermentation. Another name for cellular respiration is aerobic respiration, or we could also say it's the oxidation of glucose. Well, if I keep on thinking about it, maybe I can say that it's also a catabolic pathway. In this chapter, we're going to talk about the 4 steps of cellular respiration: glycolysis, pyruvate oxidation, the citric acid cycle, and the electron transport chain. We're also going to talk about fermentation too, and that fermentation is basically just glycolysis. We'll compare the amounts of ATP produced between both aerobic and anaerobic respiration. So, before we get started, isn't it true that all life requires energy, and at the cellular level, the form of energy is in the form of ATP? So this energy is going to come from glucose, and glucose is going to use the bonds broken from the catabolic process of breaking glucose down into CO2. That energy that's released in all those chemical bonds will be used to add a phosphate to ADP to make ATP. So ADP represents adenosine diphosphate, and that means a 2-phosphate tail. And when we connect that 3rd phosphate, it becomes adenosine triphosphate. And keep in mind, this is an activated nucleotide. So most cells generally contain enough ATP to sustain energy for about 30 seconds. Now, the reason why cells don't store ATP because it's unstable, and that's why cells have to make it all the time. In other words, that's why we're breathing all the time, is because ATP is unstable, the cells can't store it, so we need it generated as we use it. So cells obtain glucose to make ATP. In other words, to add that phosphate to ADP to make ATP. Plants produce glucose during photosynthesis, and then other organisms feed on that food, and that's how we get our energy. So we can do it directly or indirectly. But in any case, it's ultimately the breakdown of sugar to CO2 and water that's going to release that energy. I love this formula. Glucose C6H12O6 in the presence of oxygen produces CO2, water, and energy in the form of heat and light. So oxygen atoms are reduced to form water. So remember what reduce means. Do we accept or lose electrons? You're right. We accept electrons. So at the very, very end, we're going to see that oxygen, the terminal electron acceptor, is going to accept protons and electrons and form water. So why does it take so many steps to oxidize glucose? Well, we could just torch sugar and you could have a great big flame, but the truth is to harness all that energy, it takes a stepwise procedure to break all of those bonds in glucose, C6H12O6. So here energy is going to be transformed to be used to synthesize ATP. Fermentation also oxidizes glucose, but it's not fully oxidized. In other words, it's only the first of the 4 steps of cellular respiration. In other words, it's just simply glycolysis, and the terminal electron acceptor is some form of pyruvate, and so for that reason it really isn't fully oxidized, and for this reason only a few ATPs are produced for every molecule of sugar. I love figure 9.1 because it shows the full circle of energy. Isn't it true that the ultimate source of energy is the sun? And photosynthesis captures that sunlight and makes sugar. And that sugar is used by humans. And like in this drawing, they're actually they're rowing in boats and they need energy, so they're eating carrots. So they're breathing hot and heavy to break down that sugar to produce ATP so they can row the boats. Notice with fermentation, there is no oxygen. So small organic molecules capture the energy to produce only 2 ATPs. So what are the 4 steps to oxidize glucose or for cellular respiration? Glycolysis, step 2, pyruvate processing, step 3, the citric acid cycle, and step 4, the electron transport chain. So what happens in glycolysis? A 6-carbon glucose is broken down to 2 3-carbon pyruvates. In pyruvate processing, pyruvate is oxidized. It goes from a 3-carbon to a 2-carbon molecule, and then extra molecules are added to form acetyl-CoA. And then in step 3, acetyl-CoA is completely oxidized to CO2. So we go from a 2-carbon molecule to just a 1-carbon molecule, which is CO2. At that point, in step 3, glucose is completely oxidized. All of those electrons from the breaking of the bonds, because let's remember that covalent bonds are the sharing of electrons. So when the bonds are broken, where do the electrons go? All of the electrons will go to step 4, the electron transport chain. Or another name for this is oxidative phosphorylation. And all of those cascading electrons will be used to generate ATP. I also enjoy figure 9.2. It says what goes in and what goes out. And also notice for each of the 4 steps, it also tells you where it occurs. So let's look at glycolysis, step 1. Glycolysis occurs in the cytosol of eukaryotes and prokaryotes. Well, what goes in? Glucose goes in, and what's coming out? NADH, pyruvate, and ATP. What is NADH? We're going to find out that NADH is an electron carrier, and ATP is energy formed by the breaking of those bonds directly. This is called substrate level phosphorylation. Okay, let's go to step 2. Step 2 is pyruvate processing, and notice here it says it happens in the mitochondria or in the cytosol of prokaryotes. So what goes in? Well, pyruvate goes in, and there's 2 for every glucose molecule. What comes out? More electrons carried by the carrier NADH, acetyl-CoA, and CO2. In step 3 also happens in the mitochondria, the matrix of the mitochondria, and that's the citric acid cycle. So what goes in? Well, acetyl-CoA goes in, and what comes out? 2 electron carriers, NADH and FADH2, CO2, and ATP. And then the last step is the electron transport chain, and this happens in the inner membrane of the mitochondria or the plasma membrane of prokaryotes. So what goes in? NADH from step 1, NADH from step 2, NADH and FADH2 from step 3. They all go to the inner membrane of the mitochondria. And also oxygen goes in at this step. Oxygen and all those electrons and protons, and what comes out? ATP and water. So remember, oxygen is reduced to form water, and most of the ATPs are made in step 4. So how else can we describe cellular respiration? Well, it's a set of reactions, and it's using electrons to actually synthesize ATP. So it says there's 2 fundamental requirements of a cell. Step 1 is energy to generate ATP, and 2, a source of carbon to use as raw materials. So what does that exactly mean? Why is it that I like candy so much? Well, I eat my candy bar or my Hershey's bar for 2 reasons. I always say that I need it because when I lecture, I'm always moving my hands because I'm highly animated. But the other reason why I need it, it's not only just for the energy, I need the carbon in the candy bar to make more of me. Cellular respiration plays a central role in metabolism. And remember what metabolism is? Metabolism is the sum total of all reactions. Well, we can divide these reactions into two basic pathways, catabolic and anabolic pathways. Catabolic pathways break down molecules and often harvest energy to produce ATP. Anabolic pathways are pathways that actually build large molecules from smaller molecules, and it often requires ATP. So these two pathways are often coupled together. So what is cellular respiration? Is cellular respiration a catabolic or anabolic pathway? Yeah, you're right. It's catabolic pathway. Catabolic pathways break down a variety of molecules. So cellular respiration interacts with other pathways. Now, in this chapter, we're mainly talking about the breakdown of glucose to produce ATP. But could you substitute carbohydrates for other macromolecules? Could we put in fats or proteins to actually produce ATP? And the answer, of course, is yes. Like when you look at certain diets, like the paleo diet or other diets that exclude sugars, isn't it true that we still can produce energy? Because we can produce energy from fat, and we can also produce energy from the breakdown of proteins. This won't be on your test, but it's sort of nice to know. Someone had mentioned that they were on the paleo diet, and they had mentioned from one student to another, they mentioned that if you eat fat, that fat doesn't produce fat, but only sugar can produce fat. And I started thinking about it, and I don't know if that's entirely true, but there is a kernel of truth, I think, in that statement. So fats can be broken down, and from that you can get energy. And the same thing applies. Proteins can be broken down into amino acids. And again, when the bonds are broken, that energy can be transformed to synthesize ATP. Molecules found in carbohydrate metabolism are used to synthesize molecules. So even though we think of cellular respiration as a catabolic pathway, in other words, going from a large molecule like sugar and breaking it down to CO2, really parts along the way in each of the steps can actually be pulled away and used to build other components in the cell. So the products, the intermediate products, can be used to make things in the cell like DNA and RNA and even phospholipids for the plasma membrane. Figure 9.3 at first glance is sort of confusing, but you can see the 4 steps right in the middle of the diagram. You can see glycolysis, pyruvate, acetyl-CoA, and the citric acid cycle. In fact, that's steps 1, 2, and 3. But if you look above, you see that it's a catabolic pathway. Sugars, fats, and proteins can be broken down to make energy. But if you look below that pathway, you see anabolic pathways. Intermediates from this pathway can be used actually for nucleotide, phospholipid, fat, and actually protein synthesis. So this is a very important pathway, and you must know this like the back of your hand. So, as mentioned before, catabolic intermediates are used in anabolic pathways. The intermediate products from each of the steps 1, 2, and 3 can be pulled out, and they can be used actually to build other macromolecules needed within the cell. And this use of using cellular respiration as both catabolic pathway and for products for anabolic pathways is often referred to as homeostasis. It helps maintain a stable internal environment within the cell. I love this figure 9.4 because in essence it shows that at first glance maybe we think that cellular respiration is an easy pathway to understand, but the truth is it really is quite complex. I think this is one of the best slides. If you use this slide as a guide for each of the 4 steps to understand cellular respiration, you will do very well on your next test. You have to ask yourself, for each of the 4 steps of cellular respiration, what goes in, what comes out, what happens to the energy that is released, where does each step occur, and how is it regulated? If you know all of those answers, you're in a very good position to do well and to know this pathway forever. So when we talk about step 1, glycolysis, if we take that word apart, glyco means sugar and lysis means break. So we're breaking sugar and we're breaking it from a 6-carbon molecule to 2 3-carbon molecules. That 3-carbon molecule is called pyruvate. But in actuality, glycolysis takes 10 steps. The important thing to note is that glycolysis, to get it started, needs 2 ATPs. And at the end, it's going to make a total of 4 ATPs. But the net gain is 2. So you make 4, but it took 2 to get this first step started. So your net gain is 2. 4 minus 2 is 2. So notice that ATP is produced in this step, and we call it substrate level phosphorylation. In other words, ATP is produced directly from the breaking of bonds in sugar, and that's why it's called substrate level phosphorylation. It also yields protons and electrons that are carried by NADH, a molecular electron carrier. So the net yield of glycolysis, which happens in the cytosol, is 2 NADHs, 2 ATPs, and 2 pyruvates. And in this step, you can see that ADP is phosphorylated. In other words, another phosphate is added to it, and the phosphates here are the yellow spheres. So notice ADP plus P is ATP. Here are the 10 steps. So this is a good question. Do you need to know the 10 steps for the next exam? And the answer is no. You don't need to know each of the steps, but do you need to know that it takes 2 ATPs to initiate glycolysis? Yes, you do. Do you need to know that 4 ATPs are made? Yes, you do. And the net yield is 2. You also need to know that there's a lot of electrons and protons released. So here are the 10 steps, and for those of you interested in chemistry and the enzymes used to mediate each of these 10 reactions, please take a look at this table. So how is glycolysis regulated? It's regulated by feedback inhibition. So high levels of ATP, which is the end product of cellular respiration, if you have a lot of ATP, it's going to inhibit one of the first enzymes regulating the 10 steps of glycolysis, and that enzyme is phosphofructokinase. So if you have a lot of ATP, then that ATP will slow down or inhibit the catalytic capabilities of phosphofructokinase. So it says here phosphofructokinase has 2 binding sites for ATP. When ATP binds to the active site, the enzyme catalyzes the 3rd step in glycolysis. But when ATP levels are high, it binds to a regulatory site and inhibits the enzymes. So it's allosteric regulation, and that is what's going to stop the enzyme from continuing or working. So here it shows the secondary binding sites. Step 2. Step 2 is pyruvate processing, and this processes pyruvate to acetyl-CoA. In other words, we're going to from a 3-carbon molecule to a 2-carbon molecule, acetyl-CoA. And this is going to occur in the mitochondrial matrix. So I want you to keep in mind that mitochondria have a double membrane. There's an outer and then inner membrane. And in between, there's a matrix between these two membranes. There's a liquid. And so this is where pyruvate processing occurs. It occurs in the matrix or between the sac-like structures. So in this diagram, you can see that mitochondria have a double membrane. The outer membrane and then the inner membrane is folded like a labyrinth. And in between those spaces is the matrix. So pyruvate processing takes place in the matrix of the mitochondria, and what drives it is the enzyme pyruvate dehydrogenase. So it goes through a series of oxidation steps, and one of the carbons is oxidized to CO2. In pyruvate processing, NADH is also produced. In other words, the electron or molecular carrier NAD becomes reduced and carries protons and electrons. And then at the end of the processing, coenzyme A is added, producing acetyl-CoA. This diagram shows that pyruvate has 3 carbons, but one carbon, that carbon in red, is going to be lost as CO2, leaving only 2 carbons, which will form acetyl-CoA. And notice that coenzyme A and sulfur are added, forming acetyl-CoA. During these reactions in pyruvate processing, pyruvate, NADH, and acetyl-CoA come out. And pyruvate processing is also regulated by feedback inhibition, just like step 1, glycolysis. So when there's abundant amounts of ATP, then pyruvate processing will slow down. Step 3 is the citric acid cycle, and the citric acid cycle also takes place in the mitochondrial matrix. Now keep in mind, if we're talking about prokaryotes, everything is going to occur in the cytosol. So what happens in the citric acid cycle? Acetyl-CoA, which is a 2-carbon molecule, will be completely oxidized into 2 CO2s or 2 carbon dioxide. So now glucose is completely broken down from a 6-carbon molecule to 1 carbon, which is CO2. So what kind of energy is released during the citric acid cycle? Well, a lot of electrons are released. So those electrons reduce NAD to NADH2. They also reduce FAD to FADH2, and ADP is also forms to ATP. So there's substrate level phosphorylation in step 3. In other words, from the oxidation of acetyl-CoA, ATP is formed directly. So substrate level phosphorylation occurs in step 1, glycolysis, and in step 3, the citric acid cycle. I love this diagram of the citric acid cycle. So you can see why it's called the citric acid cycle, because there's a total of 8 organic acids, and the first one is citrate, citric acid cycle, and the last one is oxaloacetate. Now, do you have to know all of these organic acids? And the answer is no. But do you need to know that step 3 occurs in the matrix of the mitochondria? Yes, you do. Do you need to know that ATP is formed in this step? Yes, you do. And that electrons and CO2 are also released, electrons and protons, and those electrons and protons are carried away by NAD and FAD once they become reduced. And keep in mind, reduced means they accept both the electrons and the protons. Remember, when I talk about a proton, I'm talking about hydrogen. So here are the steps for the citric acid cycle. You don't have to know these for the next test. So how is the citric acid cycle regulated? Okay, it should be no mistake. It is also regulated by feedback inhibition. So when there's high amounts of ATP, then the cycle slows down. When ATP rates are low and scarce, then the cycle speeds up. So here you can see the steps of the citric acid cycle and its regulation by ATP. So that's a good question. What happens to all of the NADH and FADH2 that is reduced in steps 1, 2, and 3? So remember, NAD is reduced in steps 1, 2, and 3, and FAD is only found in step 3, the citric acid cycle. So it says for each molecule of glucose that is oxidized, the cell produces 6 CO2s, 4 ATPs, and that's directly, so that's substrate level phosphorylation, 10 NADHs, and 2 FADH2. So what happens to these electron carriers, the 10 NADHs and the 2 FADH2? Where do they go? They go to step 4. So here, this is a diagram showing all the energy that has been made. So we start off with glucose, and now it's completely broken down to CO2. And notice that we have a total of 4 ATPs, 2 from step 1 and 2 from step 3. So now where are all of those electrons going? Those that are carried by the electron carrier NAD and FAD. Sorry about that. So where does NADH and FADH2 go? Where do these two reduced electron carriers go to release all of these electrons and protons? Well, they're going to go to step 4. And this is why we breathe oxygen, because oxygen is used in step 4 to produce ATP. And that's why the electron transport chain is often called oxidative phosphorylation, because oxygen is needed to form ATP. So what happens to all of that energy, and how is the energy used to make ATP? Well, what's going to happen is that we're going to create a proton gradient, and this proton gradient is going to ultimately be used to produce ATP. So this is happening in the inner membrane of the mitochondria, in the cristae of the mitochondria. Now, if we're talking about prokaryotes, it's happening at the plasma membrane. So how does this work? How is it that we create a proton gradient? And remember, when we talk about protons, we're talking about hydrogen. Well, the electron transport chain is like a cascade of electrons. You know, this reminds me of the movie The Matrix, where you see a cascade of zeros and one, that binary code that's creating all of these artificial environments. In our case, we see a cascade of electrons and protons. And the cascade of electrons are going from higher to lower energy. But you know, again, energy cannot be created nor destroyed. That energy is going to be used to actually pump protons or hydrogens on one side of the inner membrane of the mitochondria. Now, this isn't done alone. There's other non-protein and protein enzymes that are helping in step 4. And one is ubiquinone, also known as coenzyme Q or just Q. For the upcoming test, you don't have to know these helper molecules, but in the back of your mind, you should keep this in mind. As electrons move from one molecule to another, they're going through oxidation and reduction steps. And as they move, it's like a chain, this chain of molecules. And as the electrons move along this chain, they start to slightly use energy. In other words, they go from high potential energy to less potential energy. Well, where is that energy going? It's going to be going to 4 protein complexes that are embedded in the inner membrane of the mitochondria. And so that those electrons are creating enough energy to create these pumps. And what they're doing is pumping protons on one side of the inner membrane of the mitochondria. In this graph, you can actually see the 4 complexes. Complex 1, 2, 3, and 4. You can also see that electrons are being released in complex 1 and 2, and they're going through a series of oxidation and reduction steps. And as they move down this chain of different types of molecules, then that energy, they start gradually using energy, and that energy will be transformed to pump protons in each of these complexes onto the other side of the inner membrane of the mitochondria. Now notice that some of these molecules have iron. So when we talk about iron poor blood, now you understand why we need iron. We need iron because it's needed in step 4. It's needed in the electron transport chain to actually make ATP. So it tells you the enzymes that are used in each of the complexes, but to be honest, this information will not be included on the test. So what is the role of the electron transport chain? As these electrons go from complex 1 to 2, 3, and 4, we know that there's a series of molecules there, and they're going through a series of oxidation and reduction steps. And as they lose their energy, that energy will be transferred to pumps, and these pumps will pump protons onto one side of the mitochondrial membrane, creating a proton gradient, and this is called a proton electrochemical gradient. So in this diagram, you can see that complex 1, 3, and 4 are actually like little pumps, and these pumps are pumping out protons on one side of the inner membrane. And so now we've established a proton gradient. And this energy came from the movement of all of those electrons that were released from NAD and FAD. Now, how is it that energy is formed? Well, embedded in the inner membrane of the mitochondria is another enzyme, and this enzyme is called ATP synthase. So now let's look at this word, ATP synthase. Well, we know it's an enzyme because it ends in ase, and synth, the prefix, means to make. So what are we making here? ATP. So now that we have this electrochemical gradient of protons on one side of the membrane, now that it needs to reach equilibrium again. In other words, there has to be a net amount of protons on both sides of the membrane. But this is the thing. The only way that the protons can cross the membrane is through this enzyme called ATP synthase. So this ATP synthase is like a turbine that you would see like at Lake Mead in Las Vegas, around Las Vegas. So those turbines, when water's going across, actually start to spin, and that's how we get energy. At the cellular level, ATP synthase begins to spin, and as it spins, it's going to produce enough energy to phosphorylate ADP to make ATP. So when we say that we're producing ATP, what it means is that we're adding an inorganic phosphate to adenosine diphosphate. It has two ATPs, and we're adding the third one. Now, this movement of all of these protons cascading through ATP synthase is called the proton motive force. The proton motive force, and this is based on the proton electrochemical gradient. And remember, how was it established? It was established by the movement of all of those electrons cascading from one molecule to another. There's an experiment in your book, but I don't particularly like this experiment, but it shows how they knew that there was a proton electrochemical gradient. Because if you have protons on one side of the membrane, then that's going to reduce the pH and make it an acidic environment. The proton motive force couples the electron transport chain to actually produce ATP. So remember, the only way that the protons can cross the membrane is through ATP synthase, that enzyme or protein embedded in the inner membrane of the mitochondria. Now, ATP synthase is a very large macromolecule, and it contains a knob, and it contains other parts that keep it stable. But this knob starts to spin very rapidly as a cascade of electrons or protons come running or cascading through from one side of the membrane to the other. In this diagram, it explains exactly what is mentioned in the previous slide. We see that ATP synthase in green is embedded in the inner membrane of the mitochondria. We actually see the electrochemical gradient of protons. We see that there are a lot of hydrogens on one side of the membrane, but not on the other. And the only way that they can cross is through ATP synthase. That knob begins to twirl as protons come cascading through from one side of the membrane to the other through ATP synthase. The rotor and the stator stabilize the enzyme, so as it's twisting, that energy will be used to form ATP. Energy to produce ATP comes from a proton gradient, also known as oxidative phosphorylation. Step 4 is known as oxidative phosphorylation because it requires oxygen, and that's why we're constantly breathing, so that it can be used in step 4. That means that oxygen is the terminal electron acceptor, because remember, energy cannot be created nor destroyed. So all the low energy electrons gravitate to oxygen to ultimately form water. Most of the ATP made during cellular respiration is made by oxidative phosphorylation. In other words, most of the ATP is produced or synthesized in step 4. Chemiosmosis is another way to describe the use of protons to produce or synthesize ATP. In your book, it says that this must have occurred early in evolution. And this is a turning point, because there would be no such thing as multicellular organisms if we did not have cellular respiration, because fermentation only can produce 2 ATPs per 1 molecule of glucose. So the theory is right now that oxidative phosphorylation, or the electron transport chain, may have evolved before other electron transport chains existed. This proton gradient, excuse me. This is one of my most favorite diagrams in the whole chapter. I particularly enjoy 9.19 because it's so informative. It gives you the 4 steps of cellular respiration. It tells you where it occurs. It tells you what goes in and what goes out. It tells you what happens to every part of the step and how much ATP is made. So notice step 1 produces 2 ATPs. Step 3 produces 2 ATPs, but step 4 can produce 25 ATPs. So 25 plus 2 plus 2 gives us 29. Now, you can read in other books that sometimes they'll say maybe less than 29 ATPs are made, or even over 30 ATPs are synthesized. But the truth is, no matter what, cellular respiration produces way more ATPs than fermentation when there's no oxygen available to break down sugar. So now let's talk about aerobic versus anaerobic respiration. Aerobic means in the presence of air or oxygen. So another name for cellular respiration is aerobic respiration. When you have an in front of a word, it means no. So anaerobic respiration means no oxygen for cellular respiration. Anaerobic respiration is another word or phrase for fermentation. As you can see from step 4 that oxygen is the most effective electron acceptor because it's very highly electronegative. So there's a huge difference in potential energy between glucose and oxygen, and that difference creates the formation of a lot of energy. So therefore, aerobic organisms grow and reproduce much faster than those organisms that rely on fermentation. Okay, so the last part of this chapter is, what is fermentation? Fermentation means it's the breakdown of sugar in the absence of oxygen. So it's basically step 1 glycolysis. So a 6-carbon glucose is broken down to 2 pyruvates. Some form of pyruvate will collect these electrons and either produce lactic acid or alcohol. And only 2 ATPs are made. And this is occurring in the cytosol regardless of whether it's a eukaryote or prokaryote. So fermentation actually reduces NAD to form NADH. And that release, or the breaking of the bonds to release those electrons, will be used, that energy will be transferred to make ATP for substrate level phosphorylation. This figure is pretty interesting. So this is what it's saying. If oxygen is present in step 2, then cellular respiration will occur. But if oxygen isn't available in step 2, that is for pyruvate processing, then fermentation will occur. So there's 2 products in fermentation. Fermentation will either produce lactic acid or lactate, or it will also produce alcohol. So keep in mind, fermentation, it's the breakdown of sugar in the absence of oxygen, and it's basically step 1, glycolysis. In this example here, in figure 9.21a, we see that glucose is broken down in glycolysis to 2 pyruvates. Well, when those bonds are broken, the electrons are picked up by NAD and it becomes reduced to NADH. But what happens is that 2 ATP are also made. It says, but there's no intermediate. So pyruvate accepts the electrons from NADH. There is no step 4 for all of the electron carriers to go. So the electron carriers actually release the electrons and the protons to some form of pyruvate. In this case, it's making lactate. You can also have alcohol fermentation, and the idea is the same, but instead of making lactic acid or lactate, alcohol is produced. And in this drawing here, you can see that NADH releases electrons and protons to a form of pyruvate, and in the end, this will produce ethanol. Fermentation is much less efficient than cellular respiration. I promise you this will be a test question. It produces 2 ATPs per 1 molecule of glucose as opposed to 29 ATPs produced in cellular respiration. Now, your book mentions that some organisms can switch between aerobic and anaerobic respiration, and these organisms are called facultative anaerobes. Facultative anaerobes. So they can do either or.